The overall goal of this procedure is to induce DNA damage in a subnuclear region of a cell using a confocal fluorescent microscopy laser.
This method can help answer key questions of DNA repair, such as how proteins are recruited and retained at sites of DNA damage. The main advantage of this technique is that it allows for real-time visualization of the protein of interest, so that a profile of recruitment and retention can be built.
Begin this procedure by placing a chambered slide containing the cells of interest in a stage-top incubator maintained at 37 degrees Celsius and 5% carbon dioxide. Register image fields using an encoded automated microscope stage. Select a recognizable feature of the culture vessel, such as the barrier between wells, collect an image, and record the XY location. This will allow for alignment and registration of the XY locations of selected fields following sample preparation. Once image registration is complete, select a field for micro-irradiation and focus the sample. For cells expressing fluorescently labeled proteins, select the focal plane with the maximal nuclear cross-section in the fluorescent channel of interest.
Focusing on the maximal cross-section of the nucleus is crucial because it ensures that the focal plane is centered on the nucleus for monitoring recruitment of the protein of interest to the site of induced damage.
Select a clear feature within the nucleus, such as a nucleolus, and move the focal plane up and down while observing this change in appearance. The true focal plane will lie within the transition from light to dark. To focus the sample, select the focal plane in which the selected feature has the sharpest contrast. The next step is to register the position of the field of interest. Create a